Okay, so this is the tail end of chapter 2. This is part 4. We're going to be talking about two different things in this last part. So we're going to start off with finishing our discussion of water, where we talk about pH. And then we will switch gears, and I'm going to introduce you to carbon and organic molecules. So kind of two different topics in this video. Okay. So to start off with, again, we're going to talk about pH and acids and bases. And so pH is going to be a measurement of the hydrogen ion concentration in a solution or a substance. Okay, so what is a hydrogen ion? We've looked at hydrogen. A hydrogen ion is an H+, so it's literally a proton. Okay, and again, it's written as H+, so you'll see down here, you can actually see H with a plus sign as a superscript to the right. That is a hydrogen ion. So when we measure pH, we're actually measuring the concentration of those ions in a solution. And what this will measure, what it will tell us is how acidic or basic, or if the solution is neutral, it will tell us that. So it measures the acidity or the alkalinity of a substance. And over here on the right, I have a picture of litmus paper. Maybe you've used this before. Litmus paper is used in the lab, again, to measure pH. And we've got other technology as well that we can use, but these are, it's paper that you just kind of dip into a solution and it will change color based on whether it's a basic alkaline solution or an acidic solution or neutral. Okay. And so that brings me to what an acid is, and I'm going to show you the pH scale on the next slide. But before we look at that, I just want to make sure you understand what an acid is and what a base is. So an acid is going to be something, a substance that is going to dissociate, which means to come apart. It's going to dissociate in solution to yield or give off hydrogen ions. Okay, so those H+, those protons. It will also give off an anion, but again, what we're measuring is the hydrogen ion concentration. So here is a reaction, okay, kind of showing this. So an acid is going to yield a hydrogen ion and a negatively charged ion, an anion. Here's a great example of an acid. This is hydrochloric acid, which is HCl. This will come apart or dissociate in solution to give off, again, protons and a negatively charged chloride, anion. Okay? So what an acid does is it increases the concentration of protons in a solution. A base, on the other hand, is going to do just the opposite. So it is going to disassociate in solution to yield a hydroxide ion, which is an OH negative, and yep, here it is right here, OH negative, as well as a cation. Okay, so what this is going to do is it's going to increase the concentration of hydroxide ions in solution. Or we could say that a base will decrease the amount of hydrogen ions in a solution, and they do that by accepting hydrogen ions. Okay, so here's an example of a base. This is sodium hydroxide, so Na is sodium, and this OH here is hydroxide. If we were to put this into solution, it would dissociate or come apart, giving off a sodium cation and a negatively charged hydroxide ion. This hydroxide ion could then bind to protons, H+, forming water, and what that would do is bring down the hydrogen ion concentration. Okay? Okay, so here is the pH scale, and let me move my camera. Okay, oh, it got cut off here at the top. Oh, okay, so the pH scale runs really from 0 to 14, even though this is cut off a little bit here. And that's okay, not a big deal. And so a solution that's, highly acidic is going to be down here in this region, 0 to 2 kind of. A solution that's neutral is going to be 7. And a solution that's basic is going to be above 7. And a really basic or really alkaline solution is going to be like 12 to 14. Okay, so let's go through this. So again, a neutral pH is a pH of 7. At a pH of 7, we say that the concentration of hydrogen, H+, and hydroxide, OH negative, are going to be equal to each other. Okay, a great example of a neutral solution would be distilled water. Okay, or your blood. Your blood is relatively neutral. It's about a pH of 7.4, so it's relatively neutral. On the other hand, an acidic solution is going to be anything under a pH of 7. Okay, so again, here on my little graphic, if this is a pH of 7 where I have distilled water, if I go below 7, I'm going to get increasing acidity the farther down the pH scale I go. Okay, and so the farther down I go, the higher the hydrogen ion concentration compared to hydroxide ion concentration. Okay, so remember again, acids give off and increase the number of hydrogen ion concentrations in a solution. And so that's what that means. The more acidic, the more hydrogen ions are going to be in that substance or solution. Here's some examples for you, and some of these are on this picture and some of them are not. Vinegar. Okay, vinegar is a common, is it on here? It is not. So vinegar is a very common thing that we have in our kitchens, right? The scientific name for vinegar is acetic acid, and it has a pH of about 2.4. Okay, we use vinegar in things like salad dressing. I use it to clean. Okay, we clean out, our humidifiers and our coffee pot with vinegar. I clean my dishwasher and my washing machine. Okay, it's great for that. Something else that's acidic is lemon juice, right? I put lemon juice in lots of stuff, my water. Lemon juice is citric acid, that's the acid, and it has a pH of about 2.3. Battery acid. Inside of batteries, that's sulfuric acid, and that's really acidic. That's a pH of about 1. Inside of your stomachs, you have gastric acid, and gastric acid's pretty acidic as well. Gastric acid has a pH of about 1 to 3. Okay, so even inside of your bodies, your blood is, yes, neutral around a pH of 7. However, there's going to be areas in your body that are going to be acidic and basic. So inside of your stomach is one of those compartments that's going to be, it's going to have a pH lower than 7. It's pretty acidic in there. Okay. On the other hand, a basic solution is going to have a pH above 7 on our pH scale. And it's going to be just the opposite here. So as we go above 7, the concentration of hydrogen ions is going to decrease, while the concentration of hydroxide ions will increase. Okay. Some examples of basic, we can use the term alkaline as well. So basic and alkaline, kind of are used interchangeably. So in a basic solution, some examples of basic solutions, household ammonia is pretty basic. It has a pH of about 11 to 12. Sodium hydroxide it's very basic, so it has a pH of about 14. Baking soda, you've probably all used baking soda, has a pH of about 8 to 9. And there's other examples on here as well. So soapy water, bleach, milk of magnesia, seawater's slightly basic. Okay, so you should be a little bit familiar with this pH scale. Okay, this is just a nice picture kind of showing hydrogen versus hydroxide ion concentrations in neutral versus basic versus acidic solutions. So let's look first at our neutral solution here in the center. You will see here, if you count them up, there's a pretty equal number, maybe exactly equal number of OH negatives, so hydroxide ions, and H positive, which are hydrogen ions. Okay, so we would say since those are equal, this is going to be a neutral solution with a pH of about 7. Let's go over here to my right. We'll look at the acidic solution next. So the acidic solution, again, if we were to count those up, what we would find is there's a greater number of hydrogen ions compared to hydroxide ions. So there's definitely a concentration difference there. And so therefore it's an acidic solution. Remember, an acid is going to be something that dissociates to increase the concentration of hydrogen ions. And so that's what we see here. The basic solution is over here on the left. And so it's the opposite of the acid, right? So there's going to be a greater number of hydroxide ions compared to hydrogen ions. Remember, a base can either give off hydroxide ions to increase that concentration, or it could accept hydrogen ions to decrease that concentration. Either way. Okay. Lastly, I want to mention buffers. Okay, so we've got just 2 slides on buffers, and let me make sure that you can see the words. Okay, so buffers, these are going to be substances that minimize fluctuation in pH. Okay, so when an acid or a base is added, this could happen in your body, it can happen in the laboratory. We want to make sure that we control fluctuations in pH, especially inside of living organisms, right? So if pH gets out of whack, your cells are not going to like that. Okay, you've got enzymes inside your cells that can only function in a very limited, narrow pH range. And so therefore, we want to make sure that we keep everything in that range. Okay, so what is a buffer? Again, a buffer is going to be a substance that's going to resist or minimize changes or fluctuations in pH when an acid or a base is added. Okay, in the lab, we use buffers to help maintain stability, pH stability of a solution, preventing it again from becoming too acidic or too basic. So again, in the lab, in the body. Buffers are commonly composed of a weak acid and its conjugate base, or a weak base and its conjugate acid. Okay, and I'm going to show you an example on the next slide. Again, buffers are important in biological systems, so living organisms, or in ecosystems in general. And again, we use them in the laboratory and in industrial applications. Okay, here's an example. And so this brings us back to the beginning of chapter 1 when we talked about characteristics of living things. We mentioned homeostasis. And so this is a great example of homeostasis. So in humans, so us, again, the pH of our blood is relatively neutral. So it's at about 7.4. And it must be maintained, right? We can't get too acidic or too basic. We've got to stay at about a 7.4. And so inside of our blood, there's going to be a substance that's going to help us to stay in that narrow range. And that is this buffer. Okay, so I wanted to provide you with an example. This is called the carbonic acid bicarbonate buffer system. And again, this is going to be found in humans, and this is going to be a buffer. It's a substance, again, that's going to help us to maintain our blood pH at about a 7.4. And so here's a reaction. Over here on the left, we have carbonic acid. Okay, so it is an acid, which means that it's going to donate, it's going to dissociate and donate a proton. And when that happens, so if we go to the right, the result of that is bicarbonate, which is here, and this hydrogen ion. And so it's an acid, it releases a hydrogen ion, and that's going to increase the concentration of hydrogen ions in our solution. Notice the 2 arrows, and so this reaction is reversible. And so bicarbonate is a base. So there's that conjugate base, right? Carbonic acid is the acid, and its conjugate base is bicarbonate. And so it's a base in that it can accept protons. And so it will accept protons and will move to the left, reforming carbonic acid. Okay, when will it do that? So carbonic acid will dissociate when there is a rise in our blood pH. Okay, and so if there's a rise in our blood pH, if we're going above 7.4, we need to bring that pH down so this acid will dissociate, increasing hydrogen ion concentrations, and that's going to bring our pH down. The reverse happens if there's a drop in pH. So if our blood becomes too acidic, the base here, bicarbonate, is going to work. It's going to grab hydrogen ions, and we're going to go back this way to form carbonic acid. Okay, so again, this is a homeostatic mechanism. It's a buffer system to help to maintain pH in blood. Okay, so that's all I'm going to say about pH and acids and bases. And again, for the second part, the last part of this chapter, I'm just going to introduce you to carbon. Okay, and in the next chapter, chapter 3, our focus is going to be on carbon-based macromolecules. And so we have to understand what carbon is, where it is in the periodic table, how many protons it has, how many electrons it has, and what that means. And so we'll spend a few minutes doing that, and then I'll start to introduce you to some organic molecules. So first of all, our cells, right? We're made of cells. Our cells are made of organic macromolecules, which include proteins, nucleic acids, carbohydrates, and lipids. And those 4 organic macromolecules will be the focus of chapter 3. We will also spend a lab on this material, and you'll actually build examples of proteins and carbohydrates and lipids, and you're going to just draw. You don't actually build a nucleic acid, but you'll draw one. Okay? When you see the term organic, I want you to think carbon. Okay? So organic molecules, organic macromolecules are going to be carbon-based. They're going to have what we call skeletons of carbon. So carbon, we've already seen carbon, right? Carbon is number 6 on the periodic table. There are a couple of isotopes of carbon, so its atomic mass is going to be 12.01. Remember, that's going to be an average of the isotopes according to their abundance on the planet. Okay, here is a Bohr model or electron shell diagram of carbon. You can see it has 2 electrons in the first shell and 4 lone unpaired electrons in its second shell. Here is the Lewis structure, the Lewis dot structure for carbon. And so this shows the 1, 2, 3, 4 electrons in that second shell. Those 4 electrons, remember, are going to be called valence electrons. Those 4 electrons are going to make carbon very versatile. Carbon can bond to 4 different things covalently. Okay, when we talk about carbon, you're going to think covalent bonds, sharing of electrons. Carbon can bond to other carbon atoms. Carbon can bond to oxygen, hydrogen, nitrogen, can bond to functional groups, which we'll talk about in just a second. Carbon can form single bonds, double bonds, and triple bonds. Very, very versatile. You can end up with straight chain molecules of carbon and branched chain molecules of carbon and cyclical molecules made of carbon backbone. And again, you're going to be building all of those in lab. I'm just going to introduce you to some of those here today. Okay, and so I want to introduce you to the hydrocarbons. And again, in the next chapter, we'll talk about more biologically relevant organic macromolecules, but for now, I'm just going to introduce you to hydrocarbons. So when you hear hydrocarbons, I want you to think carbon and hydrogen. And sometimes students confuse this because you see hydro and you think water. There's not going to be oxygen in these. We will add oxygen. That's going to come in functional groups, but a hydrocarbon is just carbon and hydrogen. There's going to be 2 series of hydrocarbons that we're going to build in the lab and that we'll talk about here. They're called the alkanes and the alkenes, and we'll talk about those in just a second. I want to just, I mentioned this, but I want you to make sure that you know that carbon can form straight chain molecules. So carbon, 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 all the appropriate hydrogens attached. It can also form ring structures. And so here's some examples of ring structures. And you're actually going to build this one right here in the lab. This is cyclohexane. Okay, if you count up the number of carbons here, there's 6. So this it's actually in the middle of the word, hex right here, that means 6 carbons. This one is cyclopentane. Pent means 5. Okay, you won't build benzene or pyridine in the lab, but they're there for you. This is just to show you that we can make ring structures, and sometimes those ring structures have all single bonds, which you see here, and sometimes they have some double bonds, and look, they can even contain other molecules, or other atoms, sorry, like nitrogen. Okay, we're going to talk about these on the next slides. There we go. Okay, so hydrocarbons, just carbon and hydrogen. The first series of hydrocarbons that we're going to look at, and again that you're going to build in a lab, are going to be the alkanes, okay? When you hear alkane, okay, again, these are hydrocarbons made up of just carbon and hydrogen. The suffix here, ane, is going to tell us that these hydrocarbons are going to be saturated, which means that there's going to be all single bonds between carbon atoms, and the maximum number of hydrogens will be bonded to those carbons. Everybody's going to be full and satisfied. There's no double bonds in these molecules. They're all single bonds. The formula for alkanes that you're going to need to know is CnH2n plus 2. So what does that mean? For every carbon, there's 2 times plus 2 for hydrogens. Okay, so here's some examples down here. Here's the first 4 that you'll actually build in lab, and in lecture I'm going to ask you to do some of the drawing as well, so you get kind of introduced to it. Methane, this prefix meth means 1 carbon, and ane tells us it's an alkane. Okay, so if meth means 1 carbon, and I were to use this formula, N equals 1. So it would be C1, which we don't actually draw the 1, we just know it's there. And then H2N plus 2, so 2 times 1 is 2, plus 2 is 4. So methane, its molecular formula, would be CH4. Here's methane's structure, right here. So you have a carbon in the center, right, bonded covalently to 4 different hydrogens. And so carbon has 4 valence electrons, and each hydrogen has 1. And so we say that all of these atoms, they're full, okay? Carbon meets the octet rule, and we know that hydrogen, it only needs 2 to be full. If we go down, the prefix F means 2 carbons. So again, if I use my formula, N equals 2, so we have C2, H, 2 times 2 is 4 plus 2 is 6. Okay, here is ethane. So this is the name, this is the molecular formula, and then here is the structure. Okay, and I'm going to be asking you to write structural formulas in lecture, and I'll actually show you that a little bit. In lab, you're going to be doing that as well. Okay, so here is ethane. 2 carbons covalently bonded to each other by a single bond, and each carbon will be bonded to 3 hydrogens. Okay, and so each carbon is bonded 4 times, and each hydrogen is bonded just once. Okay, the next in the series is going to be called propane. Prop means 3 carbons. So again, I'm going to use my formula, N equals 3, C3H8. Bute means 4 carbons. So butane, again, I'm going to use my formula, C4H10. Okay, so I've got name, molecular formula, and then again, we have structures over here. Okay, so those are the alkanes. The alkenes, here's the difference. So they're still hydrocarbons. We call them unsaturated because there is going to be a double bond between 2 of the carbon atoms. And so therefore, they're not bonded to the maximum number of hydrogen atoms. Okay, we could break that bond and add hydrogens, which is actually, if you've ever read in a food label, if it says like partially hydrogenated soybean oil, something like that, that means that that oil did have double bonds in it, and those double bonds have been broken and hydrogens have been added. Okay. We'll talk more about that in lab as well. This is the molecular formula for alkanes. So CnH2n, we drop the plus 2, okay, because we have a double bond. There is no such thing as methene because we need 2 carbons and meth is just 1 carbon. So the first one in this series is ethene. So again, that prefix eth means 2. We plug that 2 into here, N equals 2. And so the formula for ethene is C2H4. And here is the structure of ethene here. So again, we have 2 carbons double bonded to each other, and then each carbon is bonded to 2 hydrogens as well. Okay. The next one is propene. Prop means 3, so C3H6. And last, which is what I have right here, is butene, C4H8. Okay, and so this is a great structural diagram. And this is what you're going to have to draw for me in both lecture and lab. Okay, so you can see the 4 carbons, and the second and third carbon happen to have the double bond here. The double bond could have been here or here, and we'll talk about isomers in just a second. Okay, but if you count up the number of carbons and the number of hydrogens, it's C4H8. Okay, that brings us to isomers. And we're close to being done. We've got just 2 more slides. So isomers, okay, this is going to come into play when you start to build things in the lab. Isomers are going to be molecules that have the same molecular formula, but they have a different arrangement of atoms. And because of that, they can display different physical properties like melting point and boiling point, and they can also have different chemical reactivity. They may react differently. They may also have different biological activity, so they may behave differently inside of biological systems, inside of organisms. So here's the 3 types, and there's pictures of the 3 over here. The first is structural. So let's start up at the top. You're going to actually build this in the lab. So we've got butane, C4H10, and we have isobutane, so it's isomer. And so what's the difference here? If you count up the number of atoms, the number of carbons, and the number of hydrogens, they're the same. The difference is how the atoms are arranged structurally. And so this is, regular butane is just a straight chain of carbons. Where isobutane, carbon 2, so carbon 2 over here, is going to have what we call a methyl group, a CH3 group, coming off of it. And there is the last carbon here. So instead of going carbon 1, 2, 3, 4 in a straight chain, this has a branched structure. So structurally, it's different. The second type of isomer is geometric isomers. So geometric isomers, we're talking about a double bond, okay? And so here's an example. This is called cis-2-butane, and this is called trans-2-butane. And so let's take a look here. Again, both are butene, there's a double bond. Okay, so if we count up the carbons and the hydrogens, they're equal. They have the same molecular formula. Here's the difference. So the double bond is between the second and third carbon in both cases. Okay, but if you look here, this CH3 and this CH3 are both on the same side of the double bond. So we say that they're in cis. This is the trans configuration. So the CH3 and the CH3 are on opposite sides of the double bond. Okay. When you build these in lab, you're going to see that that double bond locks the molecule in a plane. And so those molecules they don't just spin around. They aren't capable of moving. They're locked in that plane. So they're locked in either the cis position or the trans position. So those are geometric isomers. The last, these are called enantiomers. And enantiomers are non-superimposable mirror images. So like your left and right hand, okay? They're mirror images, but there's no way to superimpose them to get exactly the same molecule. And so here's a great example. So we have a central carbon, and that carbon's going to be bonded to 4 things. It's bonded to a hydrogen, a fluorine, a bromine, and a chlorine. That bonded to the same thing over here, but the arrangement of those atoms is different. Okay? We call them the L and the D isomer, and that has to do with how they rotate plane polarized light. Don't worry about that. Just know that they are mirror images of each other. Again, if you were to build these in the lab, you would see how you can't, they're not superimposable. Okay? Enantiomers, importantly, the drug that's used to treat Parkinson's disease is the L form of a specific enantiomer, and that's the only form that will work. The D isomer does not. It doesn't actually bind to cells. And so again, there's different biological activity. Some, especially with drugs, some drugs will only work in one form. Okay. Okay. Ooh. Skip the last slide. This is the very last slide. So we're just going to take a peek at functional groups. And again, you'll be drawing some of these for me in lecture, and you'll be building some of these in lab. So a functional group is a group of atoms that occurs within a molecule that's going to give that molecule specific chemical properties. Okay, and here's our list. And we're going to be building just a couple of these in lab. We will build the hydroxyl group, which is also called the alcohol group. The R here stands for whatever molecule this is attached to. The hydroxyl group is made up of an oxygen covalently bonded to a hydrogen. And a hydroxyl group, because of this oxygen, will give the molecule polarity. Okay, we'll have a partially charged region because of this oxygen. You'll also build a methyl group in lab. And so a methyl group, we've already looked at methane. A methyl group is just lacking one hydrogen. So instead of being a CH4, it's a CH3 group. And this can be added right to another molecule. We will not build the carbonyl. The carbonyl is a carbon double bonded to an oxygen that falls in the middle of a molecule. So these R's are, other parts of the molecule. You will build a carboxyl. The carboxyl and the carbonyl are kind of similar. So again, we have some molecule that this is attached to. And then we have a carbon double bonded to an oxygen. And that carbon is also going to be single bonded to an OH. Okay. Molecules that have a carboxyl group are acids. So this is also referred to as the acid group. What will happen is in solution, this will release this proton. So it's an H plus donor. And so again, that's the definition of an acid. It increases the hydrogen ion concentration in the solution. So that's exactly what the carboxyl group will do. So again, you'll build this in lab. You'll also build an amino group. So an amino group is going to be, again, attached to some molecule. It's going to be nitrogen covalently bonded to two hydrogens. Okay. The amino group is considered to be a base because this nitrogen could pick up another H plus to become NH3 plus. Okay. The phosphate group you will not build in lab, but I just want to show this to you here. This becomes important when we talk about DNA structure. So the phosphate group is going to be a phosphate, double bonded to an oxygen, and then single bonded to two OH groups, and then bonded to one oxygen here. Okay. The phosphate group can also act as an acid, releasing protons, H pluses, and it would end up overall with a negative charge, okay, because of that loss. So this would end up as an O negative. And the last group, again, we're not building this one in lab, but you'll see it when we look at proteins. This is the sulfhydryl group. So again, you have some molecule bonded to sulfur, which is covalently bonded to a hydrogen. And because of the electronegativity of sulfur, this also tends to be polar. So it gives polarity. Okay? Okay, that's it for this last part of the chapter.